Good afternoon, and I welcome you all to DAE Iconic Week celebration of Azadi Ka Amrit Mahotsav program. I also welcome the members who have joined online from various campuses. So, in fact, this Azadi Ka Amrit Mahotsav we are celebrating for last 1 year, and specifically this week we have several lectures from eminent speakers from various fields. So today we have a distinguished speaker, Professor Ram Viswakarma, and he would deliver his lecture in the area of life sciences using online platform. He was supposed to come, but because of some this thing, he could not come personally. So now it is my pleasant duty to introduce the speaker, Professor Ram Viswakarma. So Professor Viswakarma obtained his PhD from CDRI Lucknow. And he was a postdoctoral fellow at the University of Cambridge, and he worked on biosynthesis of two important biomolecules. One is cyanocobalamin, that is also called vitamin B12, and also the hemoglobin. Professor Viswakarma is currently a distinguished scientist with the Council of Scientific and Industrial Research, New Delhi, and also associated with CSIR Central Drug Research Institute, Lucknow. He served as the director of CSIR Indian Institute of Integrative Medicine, Jammu from 2009 to 2020. Prior to joining this CSIR IIM Jammu, he served as a vice president, medicinal chemistry at Piramal Healthcare Limited at Mumbai from 2005 to 2009. He also worked as staff scientist at the National Institute of Immunology, New Delhi from 1993 to 2005. So Professor Viswakarma has core expertise in medicinal chemistry, new drug discovery, glycobiology, preclinical and clinical development, and also in drug regulatory affairs. Professor Viswakarma has discovered a number of drug candidates and led them to the preclinical and clinical development stage. He has over 350 original publications in internationally reputed journals and over 75 patents to his credit. He is an elected fellow of National Academy of Sciences of India and also received several awards. Notably, he received the Sun Pharma, formerly known as Ranbaxy, Research Award of 2014 for his outstanding contributions in pharmaceutical sciences. In addition to the scientific contributions, he has led a number of societal and industrial missions such as CSIR Aroma and phytopharmaceutical missions, CSIR Innovation Complex at Mumbai, DST TBI, Jammu, and industrial biotechnology parks in Jammu, Srinagar, and biopharma and aroma entrepreneurship across the country. He has also been an advisor to pharma companies and the World Bank. He has served on a number of high-level committees of CSIR, DBT, DST, MOES, MOEF, and research councils of scientific institutions. So today the title of his lecture is Opportunities in Life Sciences Research, How New Biology is Driving Discovery of New Therapeutics. Now I request Professor Ram Viswakarma to deliver his lecture. Professor Viswakarma, please, you may start your presentation. Thank you.
Let me share. I'm trying to put my presentation. Cancel. So thank you, Professor Ghosh, for kind introduction. First of all, I will apologize that. Can you hear me?
Yes, sir.
Yeah.
Are you able to share your screen?
Yeah, just can you help me with first I will put up that. So I go to share, then
Application. First, you have to open your PPT file, whatever you want to share.
yeah, that is open. Yeah.
Then go to share and click on application.
Click on just a minute. Let me do this again. Share, application, and then window.
And window
Okay.
and
Uh,
PPT, select PPT and share button will appear.
share.
Yes, sir. Now go to your PPT slides. Don't stop. Just
Okay, PPT. Where is this my PPT?
minimize this browser.
This browser. And I go here. Can you see? Can you see?
No, sir. No, sir. Sir, you have selected browser, I think.
Okay, just I'll go back again. I am not very well versed with the.
Click on stop sharing and then share again.
Just a minute. So stop sharing. Application.
Sir, desktop screen रहेगा शायद. Select screen.
Just a minute. Share screen. Okay. Window.
And share. Click on share.
Yeah. Can you see now?
It's coming, sir.
Okay.
Yes, sir. Yes.
Can
Make
you
it.
see? Okay. It's okay now?
Yes, sir.
Yeah. So
Sir, can you make it full screen?
it is full screen from my side. Maybe it will come. Is it full screen?
No, sir. Make it in slideshow mode.
Is it from my side? It is slideshow mode. Maybe I'll do this again, and so then do this again. Now,
Sir. Sir, on the keyboard, press Alt Tab key.
Alt
And Tab simultaneously.
then I'm using Mac.
Oh.
Okay. Just a minute. On my screen, I can see the slideshow mode. View slideshow. How about now? How is this now? Hello?
Sir, it is not coming.
Just a minute. So I will make it.
Mr. Viswakarma,
Yeah.
can you share your presentation to my HBNI email or my Gmail so that we will project it from here and you can present from your side?
Okay, I can send it right away. Yeah.
Yeah, so you have my Gmail, this HBNI email, na, sunilg@hbni.ac.
Yeah, I'm just sending it. Okay. Reply attached.
जीमेल को फिर भेजना पड़ेगा उसको, जीमेल उसको नहीं है.
This is 30 MB, so this will, you have to give me Gmail. Okay, can you give me your Gmail? Hello? Hello? Can you hear me?
Professor Viswakarma, why don't you make it a PDF file?
No, so what no, if you give me your Gmail, I'll send you on your Gmail.
So my Gmail address is ghsunil,
Just a minute.
GHSUNIL, SUNIL,
Sunil, okay,
and number 60, 60,
60 at,
@gmail.com.
just I'm sending you.
Let us see.
Yeah. Because this is a bigger.
करना है मेल आईडी खोलो.
It will come in Google Drive link. Okay, so I've sent it. Hello, can you hear me?
इसमें yeah, someone is trying to sign in your account from nearby device. Yes. Yeah. ये है ना 50150 877-466. So click करो. Yes. Yeah. Yeah. Professor Viswakarma, you have to give the permission to access your file because you have sent the file, na? So you will receive one email.
Oh, okay. Permission.
Yeah, Google Drive.
Email from where? It's on my Gmail.
Your Gmail only.
Oh, this is.
You will get an email letting you know if your request was approved. So you have to approve the request so that I can download it.
Normally, I don't get it. You got the Google Drive?
Google Drive is come because when I'm trying to download, it is asking for approval.
Oh. Normally, it never asks. I don't know what's happening. I've not got any.
Which email you are sending, now you have to approve from that. Huh?
Send. Just a minute. Yeah, it has gone to you. Otherwise, what can I do? I can make that slightly bigger. My presentation. Let us see how it comes. Okay. Screen. Window. Okay. The screen.
Minimize browser.
Okay, I'm just doing this share. What I stop sharing. Screen,
Screen and
window,
window, share.
yeah, share. Okay. Now I will make it slightly bigger. How's this?
Yeah, okay, okay.
This can do. This can do. Okay.
Yeah. Yeah.
Okay. I think live with that. Can you see this?
Yeah. Sir, can you go to next slide?
Yeah.
Yes. Fine, sir.
Fine.
Yes,
Okay.
sir. You can start.
So sorry for this delay. So I was just saying that uh, I'm grateful for this invitation, and I was all set to come because after COVID, this was one good opportunity to return back to my favorite city. But last week I got viral infection, and which I'm still recovering. Fortunately, it was not COVID, so that's the reason I'm making it online presentation. So thank you for this invitation again. And I was given a topic by Professor Ghosh, which was very broad topic, uh, opportunities in life sciences research. And I then, because life sciences area is so broad that it is not possible to sort of give idea, and I given to understand that the audience mostly include young students, young faculty, and people with cross-disciplinary areas. So I made it a presentation in such a way that it is sort of a broader outline of exciting things that are happening globally in the area of life sciences research, and keeping in view of my very narrow expertise, I restricted myself to the area that I know reasonably well, which is the area of therapeutics, and where I have witnessed that in last say, 10 years, how this area has been sort of guided by new advances in biology. So that's why I've given a subtopic, how new biology is driving discovery of new therapeutics. And why I'm saying this biology driving discovery is a new phase in life sciences across the world, and particularly biotech industries. As you know, that tremendous advances are being made in developing new biologicals, new therapeutics, new vaccines. We have just witnessed during COVID that how globally, collectively, X number of vaccines could be discovered and developed in a very short period of time. So that is all possible because of very, very phenomenal growth that is happening in area of biology. And I have taken roughly 10 examples. So I will not go very deep into any topic because that will require another lecture. So I'm giving you a snapshot of the 10 areas that I have identified, which according to me are very exciting for younger people to think and young faculty to think to enter to those areas. And they are all have direct relevance to healthcare. And depending on the time availability for me, I will try to quickly go through each of those. And I have tried avoiding giving detail about my own work, our own work in those areas, but I will give you some snapshot that how we are also contributing to those areas. So these are my, can you hear me well?
Yes, yes.
Okay. So these are 10 broadly, let us see, time allowed. How many you can stop me when my time is up. So one area which fascinates me and which is a very last 30, 40 years, a lot of work has been going on, but there is a now new shift in that area, which is as we are understanding more about protein structures, their functions in biology, they are fundamental. All of us know about these kinases, GPCRs, and these have been very uh, widely focused targets. Large number of our new drugs that have been discovered in the last 10, 20 years are addressed to these set of proteins. So what is that new that is actually happening in these two areas, and which is central to actually biology? The more we are understanding about the biological system, one central message which initially was understood in terms of the genetics of biological system is now translating into sort of at the protein level, is that there is a significant amount of redundancy in biological system. And when I say redundancy in biological system is like a good housekeeper, you keep always options for emergency. So you have 10 pathways to do the same job, one job. So just in case there is a difficulty in one pathway, you can switch over to the next pathway. And that's a basically the hallmark of the biological system, how this system survives, thrives, and eventually dies. And this is the cycle of life that happens in across the sort of species. And this redundancy has been very beautifully built by evolution to make living system so robust that if you have one kind of problem, you can always, there will be other related systems which will take over the function, and then the you live basically. So these are the new ideas which are now emerging as we move forward, that there are multiple isoforms of same class of protein. And when you look at them broadly, you wonder that why I need say 13 or 14 cycling dependent kinases. All of us know these are CDKs, are very important proteins involved in the cell division. And the question is why you need, if you can do job with one, why you need 14 and 15, or you need equal number of cyclins which basically regulate them. And now those issues are getting clear, that because of the stresses, because of the diseases, because of differing requirement of the systems, sometime if there is a incapacitation of one particular pathway, the other pathway will take over. Or there are functional sort of distribution of among these isoforms. So that's one area which actually driving next generation inhibitors, next generation therapeutics. And all of you know that, in last 10, 15 years, we have at least 25 to 30 kinase inhibitors which have been approved for oncology or autoimmune diseases, which are used day to day in clinic. But most of them are actually non-specific inhibitors. And that is why most of these anti-cancer drugs are carry with toxicity associated with the activity. And that is where the problem is. So I think now the next level of challenge is that we can design and develop isoform selective inhibitors. So that's one area that fascinates me, and this is central to biology. It is very textbook information that how three-dimensional information from biology is developed from two-dimensional information, for example, from DNA, which is essentially a two-dimensional molecule. Then you go to transcribe to RNA and then go to protein. And in this process, you build huge amount of diversity in the system. And of course, there are small molecules playing important role, right from histones to RNA to protein, and then you have these hormones, neurotransmitters, all these are important. And this whole process, if you look at this, don't worry about the complexity, look at from DNA to RNA to protein in red, how the diversity is built by nature. So you have a simple DNA structure, and then you know how it is modulated by histones, multiple epigenetic factors, alternative splicing in RNA, and you keep on building diversity at the end when you reach the what we call post-translational modification, where actually you do some very simple chemistry on these proteins which are synthesized. Chemistry like you add some phosphate, glycosylation, acetylation, methylation, simple chemistry that we do in the lab. Nature is also doing similar chemistry, except the nature is using different reagents, the kind of reagent that we use in our lab when we do this organic chemistry. And the why, how it is done is that if you see that roughly very small number of genes in biological system, for example, 20,000 genes in humans, fruit fly, or worms. And these 20,000 genes reach to millions of proteins with different functions. So that is how you have, and in this in between, you have almost 37,000 microRNAs in human. And this is one area where a lot of exciting work is happening, how these things are happening. So among this post-translational modification, I will just highlight one, which is the phosphorylation, which is a central message, central sort of mechanism in cell, multiple functions of cells. And this is very simple chemistry. As you see, there are phosphorylation event that occurs on a protein backbone. So you have amino acid, you have, for example, hydroxyl group, you phosphorylate it. And these phosphorylation in mammalian systems happen mostly on serine, threonine, and tyrosine among all amino acid. And most important them are serine and threonine. Tyrosine phosphorylation is very rare. In bacteria, however, if you see, you do the phosphorylation in histidine and aspartic acid. So it's very different, clear different demarcation nature has created. And along with these phosphorylations, which are done by this protein called kinases, there are equal number of proteins which remove these phosphates after the job is done. Those we call phosphatases. So this is a very important sort of a chemical reaction that drives a lot of functions. And when there are dysregulation in these processes, you end up with the diseases. So why this simple reaction? What fundamentally it changes a particular protein structure. So as a chemist angle, if you see, if you simply put you have, for example, a amino acid in the protein backbone, which is essentially neutral. Now, if you convert this neutral hydroxyl to a dianionic phosphate group, this dianionic group will lead to recruitment of a lot of cationic arginine side chain residues from different sites of the protein. And what essentially you drive about is you bring about a conformational change in this protein structure. This is witnessed very nicely in receptor tyrosine kinase activation. For example, insulin. So when you have a insulin ligand interacting with insulin receptor during our food cycle, the receptor autophosphorylate itself. It has the catalytic activity, it autophosphorylate and send the signal down below that food is coming or glucose is coming. So that is how our metabolic activities are controlled. And when you have discontrol of those, when you have what we call insulin desensitization, you have essentially type 2 diabetes. So these are the consequences of these phosphorylation. And then these phosphorylation can happen in multiple. So one single protein, don't bother about an individual protein. It can go to large number of phosphorylation in one backbone, and it will change completely the structure of, for example, threonine 160 of CDK2 will, with the guanidinium side chain of arginine 50, and it recruits basically three these arginine and changes the three-dimensional structure. Essentially, I'm trying to say that even one phosphorylation in a large protein can bring about major conformational change, and that has a functional consequence. This is another example of the protein, which is Abl protein kinase. This is a protein which is basically mutated in large number of children's cancer, leukemic cancers. Many of you would know about this. And the first drug called imatinib, long, I think 15, 20 years ago, was developed looking at these unusual kinases. Now, these kinases, they are in resting phase and they are in active phase. And now with X-ray crystallography and cryo-EM, now people are determining the structure of the active phase to inactive phase. And now we have reached a stage in last couple of years, people have started designing inhibitors for inactive kinases, inactive phase of conformation rather than the active phase of conformation. Because active phase of conformation is when a transition has already happened. So if you can, for example, want to develop a chemotherapeutic agent, now it is clear that if you have an inhibitor which is targeting inactive conformation of protein, you are likely to have less side off target activities. So essentially now, where is the central message is here that if there are, for example, give you example of among all the kinases, I give you example of say human genome. If you see, there are 520 kinases in our body, and almost similar or lesser number of regulatory phosphatases, which are controlling most of our function in typically cell division, or all signaling is controlled by this. What we know now that if you look at the individual class of kinases, you will see that multiple kinases are doing the same chemistry, and they are isoform of each other. And there are subtle differences in terms of they do their job. For example, I gave you example of cyclin-dependent kinases or PI3 kinases, which is phosphatidylinositol 3 kinases. These are very important kinases which have 4 isoforms, and they are involved in the, for example, alpha, beta, gamma, delta. And now we know very well that alpha isoform is important in cancer. Large number of cancers, you will find PI3K alpha mutated. And that is where the first biomarker for the detection of the cancer is when you look at these isoforms of PI3 kinase. And if you see the other isoform, which is a gamma isoform, which is primarily involved in the autoimmune diseases. So now we have reached a stage is that we can design a specific inhibitor of alpha or beta or gamma or delta isoforms. That's phenomenal. And I think that is where when I say the real opportunity now is for next 10 years of life sciences is that with the help of good chemistry, structural biology, good in vitro, in vivo systems, I think we are in a position to design these specific inhibitors, both, and also in a position to design monoclonal antibodies for these particular kinases. And this is a very complex diagram where I just want to just give you a message that how these receptors which are on the membrane, say cytokine receptor, GPCR, growth factor, insulin receptor, and death receptor, these are all how integrately they are involved with large number of these kinases. And there is a possibility now that we can pick up individual kinase and start designing inhibitors, isoform selective inhibitors, so that we don't disturb this equilibrium too much and have less off-target activity. And this is I've just picked up this slide from literature that to just give you example that in last 10 to 15 years, we have at least this number of therapeutic drugs. These are all FDA approved drugs, and most of them are actually ATP competitive, non-selective. There are few examples of selective also, but most of them are non-selective kinase inhibitor. So you can imagine very well that with this stage where we are with the structural biology or functional cell biology, we have reached a stage where we can move to next phase of discovery where we can design isoform selective inhibitors. So this is one area fascinates me, and then we have done some work. And one example is that in last, I think, 7-8 years, we have been working on this molecule, IIIM290, and 2 years ago, we got approval by DCGI for phase 1 oblique phase 2 trial in pancreatic cancer. So this is where under clinical trial. And this is very specific CDK9 selective inhibitor, where this particular kinase is overexpressing in pancreatic cancer. And that is how we have selected using all kinds of structural biology, structural modeling. So I'm not going deep into this because too much data. The second area is very fascinating area, which I think after a lot of struggle, now we are beginning to see light of the day. As you see, 99% function in biological system is controlled by protein-protein interactions. One protein interacting with another protein and changing conformation, changing function, and that is like a orchestra of millions of these proteins which drives function. And the dream was that can we design inhibitors of protein-protein interaction? And the challenge is that these interactions are 100 to 200 angstrom interface. As you can see from physical chemistry point of view, that they are very large areas. Now, how can you design a small molecule to block these interactions? So this has been a major challenge, and this some people call it holy grail of therapeutic discovery. And some success has happened primarily from biological therapeutics. So there are now monoclonal antibodies people are designing where you can block these interactions. And some of these interactions, for example, the best example is like insulin receptor. And related to insulin receptor, there is a receptor called IGFR1, which if you activate, it will lead to cancer. Insulin receptor, if you activate, it leads to your control of your sugar level. So see how closely they are related. And these are essentially, there is a protein ligand and there is a protein receptor and so the interaction. So this is a very interesting area, and I've already said that for example, most of these cytokines, TNF, IL-6, and I think COVID time, you must have seen how these cytokines have done havoc in a what we call cytokine storm, where large number of these proteinaceous molecules were thrown by due to the virus induction, and they were going and blocking the receptor site and creating collapse of the lung function and other functions. So this is a very, very important area. And here, of course, I can give you one example I selected because it was done in collaboration with Institute of DAEE. So we have been doing this work almost 10 years collaboration with Tata Institute of Fundamental Research with NCBS in Bangalore and my lab in Jammu, and where we are targeting this NEF protein of HIV virus. And HIV virus basically enter our cells using down-regulating this important co-receptor, co-stimulatory receptor on the cell surface called CD8086, and that's how the virus get entry. And this is essentially an important function. And initial work was done in the lab of Dr. Shahid Zamil in ICGB, New Delhi, many years ago, where they shown the importance of this NEF in interaction in the entry of the HIV virus. So then with Dr. Mayer of NCBS TIFR, I started this program. In fact, I started this program when I was in Bombay in Piramal, and then I moved to Institute and I started this program. And now, in fact, 6 months ago, we were able to complete this process and file patents, and this manuscript now under in bioarchive. So essentially what we have done here is that we can now block the interaction of NEF of HIV virus with the CD88. These are receptor, co-stimulatory receptor on the immune cells where the virus macrophages and other cells where virus invades. So this work was done using a lot of, we set up the biochemical screen. I will not go into detail. Developed a whole lot of chemistry at my institute, and essentially a large number of these compounds were looked at. From the structural point of view, we used an NMR spectroscopy, X-ray crystallography to look at what are the binding pockets. And then essentially we ended up these scaffolds. And the most interesting scaffold, which is written here, is called Pepti Bones, which was discovered. These are the linear peptide which are produced by dying biological matter. For example, when you have a tree and the leaves fall from the tree and the trees are decaying, that decay is because of the microbial flora and fauna which is growing on the tree. So, for example, there are saprophytic fungus which degrade this biological material. So from the saprophytic fungus, we discovered that linear peptide, which are basically, they are mimic of peptides, so we call them Pepti Bones. And these showed the very, very interesting activity against this particular target. So this manuscript is in now archive, and most very soon it will appear in important biological journals. So it's one example from our own work. Third area, which is very exciting, and I think all of you must be knowing about, ultimately we are reaching now the central nervous system, the brain. And as we know, the aging population worldwide, we have a serious problem because of the problem of wealth, problem of good healthcare. People are living longer, but when they live longer, they end up with new challenges. Dementia, Alzheimer, all these diseases. So globally, there is now major push that how are you going to deal with these problems, that you have a living human being, but that human being can't think, can't communicate, lost memory, all these things are happening, and they will happen in our country also. So I think here there is an exciting area that is maximum amount of funding in the United States of America is coming to this area. In fact, 1 month ago, close to $50 billion has been committed to this particular area. And I'm sure in India also, if there is significant effort, a lot of companies, a lot of philanthropic organizations would like to take this area to next level. And this is possible because now we have much slightly better understanding of this black box of our what we call brain, which is almost 90% lipid, fat, and rest is, I don't know, we don't know much about it. And we all know the old challenges. I will not repeat them. But what has happened in last few years is, which is very, very interesting, and I see as a major highlight of life sciences, is that now we have much better understanding of the architecture and functioning of blood-brain barrier, which is the most important barrier in terms of designing new therapeutics. All of us know that 99% of the therapeutic molecule that we take, they don't go to brain. They just are not allowed. So very selected molecules are allowed to enter into CNS space. But because of structural information on these ABC transporter, many of them are ABC transporters, now we have handled that. Can we drive a small molecule which can enter? For example, neurotransmitters, they go happily. So if, for example, if you have analog of neurotransmitter, perhaps it will not go. So can we use this information and design rationally those analog of neurotransmitters? Mitochondrial dysfunction is one major area because of senescence and aging. That's how we age. Our mitochondria stop functioning with the full efficiency. And now there is a possibility that you can kickstart the mitochondrial biogenesis. You can regenerate mitochondria now in system. ER stress, which is a continuous problem of chronic neuroinflammation. I think majority of us will be suffering this, and that is how we are losing our getting more stress as we age, and that stress further causes neuroinflammation. So I think a lot of people take this very lightly, that stress and all this day-to-day life is very, very important, that it is doing long-term damage to you, and that damage is mostly because of the endoplasmic reticulum biology that is being disturbed. Now there are pathways where one can go and sort of fix this. The last area is the stem cell biology. Then now there are in vivo models, and where we can initiate, kickstart a specific type of stem cells so that you can regenerate your brain tissues and brain nerves. I think fundamental thing we were taught in textbook, that cells and nervous system do not regenerate, and that was the fundamental change that has happened. That majority of these cells in central nervous system actually regenerate. So that's a paradigm shift that has happened, and scientists are taking advantage of these discoveries that we can design. And here, very importantly, the small molecule from natural sources play a very important role, because those molecules are designed by nature to enter central nervous system because of the chemical ecological interaction of the species. So that's an interesting area that we are looking at. And so whole lot of, I will not go into detail, all this thought process, I myself has written a very kind of a quasi-scientific, quasi-metaphysical review, and where I had asked the question that why are the majority of active compounds in central nervous system domains natural products? So I think those students, those people who are interested, they can look at it, where we have categorized all these kind of structures which can become next generation questions. And some work that we did in this area, which has recently we have licensed to a company for clinical trial in the United States, is from good old saffron. We made an important discovery from saffron, that this saffron contains some compounds, very specific compounds, which activate ABC transporter of blood-brain barrier. That means you are able to efflux the amyloid beta proteins which accumulate during day-to-day activity, and you can slow the process of Alzheimer. So this is all work is patented and published. Now I'll not go into detail, but we are looking forward to doing these clinical studies with this collaboration with this company. This is a licensing that we did. The fourth area, it's very, very interesting, and I think I'm sure there will be some pharmaceutical background people in this audience. And the fundamental paradigm in last 70, 80 years of therapeutic discovery is that you identify a particular protein, which could be an enzyme or could be GPCR. So there is a well-defined pocket to which you can design your molecule. For example, if you have enzyme, then you have the catalytic domain, you design your molecule, and that molecule you can optimize to highest activity, and then say uh, that gives you what we call the paradigm of medicinal chemistry, where you optimize a molecule against a well-defined molecular target of a particular protein. Now, it's very interesting. This first work that started coming from University of Yale University, very interesting work, where they started looking at putting this paradigm upside down. That one way is to inhibit the protein molecule, the functional activity of the protein. The other way is that can we reduce the concentration of the protein itself? Because as we know, most of the proteins that are made in our system, after their job is done, they are sent to proteasome for degradation. So in nature, it's basically most efficient chemistry is there is no waste in biological system. So you utilize the proteins which have served the purpose. They go to proteasome where they are chopped off to individual amino acid, and which those amino acids you reuse and rebuild your protein again. So here the idea is that can we then reach the what we call undruggable genome? Right now, most of our effort in therapeutic discovery is restricted to not more than 5% of the total protein that are there, which we call druggable targets. But there are 95% undruggable target. When I say undruggable, that we do not know how to design molecules to inhibit or activate them. And this is where this new technology that is known as now PROTAC, proteolysis targeting chimeras. And this is a phenomenal technology. I think I will strongly recommend, and I have written some references to reviews, and student and faculty can read about it. That now, instead of inhibiting a protein and designing these molecules, what you can, if you can somehow reduce the concentration of that particular protein by tagging that protein and linking through linker to a E3 ligase. E3 ligase is a part of the sort of like a stamp, like a postage stamp. If you put E3 ligase in a particular protein, it will take to proteasome. So if you tag a particular protein that you want to reduce the concentration of that protein, because, for example, majority of the breast cancers are caused by the overexpression of certain receptor, for example, EGFR or IGFR, those receptors. Now, if I can reduce the concentration of those receptors on the cell surface of the cancer cell, I can control the tumor. I can slow down the growth of the tumor. So by this technology now there is a proof of concept. No success as yet in the clinic, but there is a proof of concept by using this technology which is described here, which is essentially a very smart way that you take a ligand, which is an E3 ligase ligand. There are several defined in the literature. You take a molecule, a small molecule will recognize your target protein, and you link through a linker, and then you give, if you give this this E3 ternary complex with hetero bifunctional small molecule takes to proteasome and destroy that particular protein. I think this is a phenomenal fundamental shift in biological system. And I can say like CRISPR-Cas technology, this is going to be an important way to deal with overexpressed protein, both in animal system, plant system, bacterial system. So I have written these three important reviews which have been published in this area, and I think I will urge students and faculty to look into this. And this is for essentially initially was for the protein which are on the surface of the cell, but now there is a technology through which we can take protein which are intracellular. So I'm not going to detail, but very recently, like last year, there was this major work that came out that we can do the same thing for intracellular protein using a particular receptor. So don't go into detail. The message is that we can now identify a protein which is overexpressed, causing problem, and I can send that protein to proteasome for destruction by very clever chemistry. So we don't have to do medicinal chemistry, we can do this protect design. Area number 5, I think, is very known to you, is mRNA vaccines, which is a remarkable success story in last 2 years that we witnessed. So what are the opportunity here? The one property, because people have been working vaccine for the last 20 years with no success, because all of you know mRNA molecule are the most labile molecule in biological system. It simply breaks down. And that was the challenge, that how it will survive in biological system. And that was a very clever chemistry that was done, that you replace a particular nucleotide in RNA sequence with a modified nucleotide. Say, for example, in this case, pseudo-uridine. Most of these mRNA vaccine, Pfizer, Moderna, are based on this particular chemistry modification, which gives them certain specificity. So I think this field is just started, born, and I think people with clever chemistry expertise, and these, it's not very difficult to synthesize these RNA sequences. There are companies who will make it for you. So there is an opportunity for next generation therapeutic, and this is going to be a fundamental way to actually deliver a lot of important vaccine and therapeutics using this approach. The second opportunity in this area is, which is virgin opportunity, to design these, what we call the carrier, lipid carrier. I'm sure all of you must have heard about these nanoparticle lipid, which were the most important lipid-based nanoparticle LNPs, which were used for mRNA delivery vehicles. And there are only few now, but I think because of the people who are in pharmacy or delivery system, is a great opportunity where you can design specifically localized targeted mRNA delivery, and you can have a patent space can be huge. So according to me, it's a very, very exciting area, and we are doing some work within CSIR. We have started a major project on this in our lab in Hyderabad, CCMB, and you will see some result coming out of that. Area number 6 is you can stop me when my time runs out, so I can stop at that point. Okay. I think, yeah, I will take another 5 minutes or so. Area number 6 is very,
so you have 20 more minutes, you can.
Okay, yeah. So this is very, very interesting area, which is happening in last, I think, 4 or 5 years only, and this is very exciting work. I'm sure all of you must have heard this new generation of cancer therapeutics, what we call checkpoint inhibitors. And these checkpoint inhibitors are basically, now we have identified the mechanism why cancer cell proliferate, why immune system cannot see cancer cell. Immune system can see any foreign cell in the body. That is how immune system is designed. Virus, bacteria, cancer. Why it is not able to see cancer cells? Turned out that it was discovered that cancer cell expresses certain molecule on its surface which block certain molecule on the immune cells and then fool immune cells so that it cannot recognize that whether the cell that it is seeing is a self or non-self. And those some of the molecule called PD1 or PDL1, and now people have been able to design antibodies against those, blocking those interaction, and then it's like you lift the curtain and immune system can see now cancer cells, and once it sees the cytotoxic T cells or CD4, CD8 cells, they go and destroy the cancer. Is a phenomenal discovery, and this biology work was rewarded by a Nobel Prize, I think 2 years ago, to a scientist who discovered these pathways. And now we are discovering a next generation checkpoint, which we call Glyco Checkpoint. I'll come to that. I don't have to tell you that these are very important in biological system. I'll come here, right here. So on the left, if you see this cancer cells and T cells, so these ligand, these interaction, they are the process through which These checkpoints are blocked. It's like there is a lock and there is a key. So cancer has discovered a key to lock the immune cell. So immune cell cannot see that there is a cancer growing up. So it remain quiet, cancer keep on growing. And now if you block that interaction of lock and key, then it is visible. So the first on the left is already a success story. The second is now evolving success story, and there is a company called Paleon Pharma in Boston, which is run by a very eminent lady in Stanford, Caroline Bertozzi. I urge you to read her work. It's a phenomenal work coming out that there are specific glycans which are expressed on the cancer cells, which block these protein called Siglex, which are on the immune cells. And now, if you can disrupt those interactions using certain proteins or a small molecule, you can actually have another checkpoint. So I think the future looks like that now we are getting into those checkpoints through which the cancer cells or virus cells or bacterial cells were fooling our immune system. And now I think we have reached a stage that we can now, instead of giving molecule from outside, we can train our immune system to destroy these cells. So you can imagine how powerful that concept is. Very, very interesting area. And I think I've given one reference below, which I can't see, but I'm sure you can see in nature immunology. Or if anybody is interested, you can write to me. I'll send you some more information on this. And we are doing some work in collaboration with a Canadian company on this aspect, where we are trying to design some conjugate molecule to block this interaction. The next area is very, very interesting, and I'm sure all of you must have heard about these stem cells for many years now. Now, what is new is now that we can now use small molecule to control the differentiation and de-differentiation and renewal of the stem cells, which is, I think, phenomenal. And this all started with this beautiful paper from Japanese scientists in 2013, published in Science. And the central message here is that all of us know about these Yamanaka transcription factors, which were discovered by Mr. Yamanaka, and he got Nobel Prize almost 10, 12 years ago for discovery of those transcription factors, which basically are responsible for differentiation of stem cell. Because all of us are born from one single cell. My father and my mother cell fuse and create this one cell, and that one cell has all the information packed that how this child will grow, and then this child will become human being. So how this ears, nose, eye, all this packed in that particular, the first cell that is generated. And that information, unpacking of information like a very complex software, is controlled by these transcription factors. Now, so that is how there is so much excitement about this field, that we can now use this knowledge and start dealing with some of the therapeutic, some of the diseases. Now, after that discovery, another Japanese scientist made this discovery that in this process, you can use actually small molecules. So what you can do, you can convert, for example, you take a skin cell, and you can convert that skin cell to liver cell or kidney cell or ear or eye, where you can use small molecule as a substitute of transcription factor. As you know, transcription factor are the proteins, regulator, and there, if you genetically manipulate them, it is irreversible. But if you do chemically, it is reversible, because if you are using a small molecule, so long as you are using a small molecule, you can do the job. But if you stop giving a small molecule, system come back to the natural state. So that's one big advantage of doing chemical biologically rather than genetically. So this is I think is a very exciting area. I'm giving example from the therapeutic perspective, that this is the future, the way it is looking at for us. And by this discovery, one thing became very clear and obvious, that there is a fundamental commonalities between development biology and stem cell biology. As I said, that most of the protein which are involved in development biology, which people have been studying using Drosophila model, all kinds of models, and which are fundamental in the development phase of life, They are also important in stem cell differentiation, dedifferentiation, and so that means if we can now reposition those molecules which are known from development biology to stem cell biology, it can be a gold mine for next generation discoveries. So, for example, hedgehog pathways, Wnt pathway. I'm sure many of you must have heard these pathways. And what we did in last almost we started a project 7-8 years on this. That we catalog a small molecule from nature. These are all natural products which are known for, some are known for 30 years, 40 years, doesn't matter. Molecule never goes out of fashion. And then we catalog those molecules to individual development biology pathway, and then systematically started testing against stem cell biology pathways in the by following the methodology described in that science paper. And it was phenomenal classification that was done, and we created this whole libraries of these compounds and tested them. I'm not going into detail, but then we ended up certain very important class of molecule. For example, we discovered new notch inhibitors, we discovered new notch activators. So the class of molecule that we discovered, notch is a very important signaling molecule pathway in development biology, also in stem cell biology. So now we are validating these in animal model, and then hopefully this is a kind of a discovery phase. And then once we have a nice collection of these molecules, you can think about where you can position them. And the area number 8 is like I call it a black box opening. Most of the immune system and immunology which is most of the discoveries are last 50 years, 60 years. It has been a kind of a non-accessible for therapeutic approaches. It was accessible first by monoclonal antibodies, and then that's why we have large number of biologicals. For a small molecule NCE, this particular area, because of the very complicated interaction of large number of players in immune system, and which is very different in the different phases. So, for example, if you are normal state, your immune system is different. When you have viral infection, your immune system is totally different. When you have bacterial infection, your immune system is totally different. And once these things go away, it goes back to. So it's very difficult to understand what is going on, how this system is. And this system has a very unique advantage, which it carries thousands and thousands or millions of the immunological memory in you. All of us are actually different individually. All of us are same biologically. You'll be surprised that I'm making this distinction between biology and immunology. This is how I like to say that all of us are identical biologically, but all of us are unique immunologically because of the unique memory, B-cell memory or other memories that we have built in terms of the infections that we encountered or my father encountered, my father's father encountered. All that is in me to protect me if I re-encounter that pathogen again. So it's a very complex area. I think it will take another 100 years or more to understand what is going on. But what has happened in the last 5-6 years, I'm watching the literature, is now this black box is opening to small molecules therapies, and particularly obviously for cancers and autoimmune disease because the money is available for research. All these areas, as you know, depend on how much money is available. And fortunately, in the United States and Europe, there is a lot of private money, philanthropic money is available for biomedical research. Almost 80% of the research funding comes from these sources rather than government sources. Unfortunately, in our country, we are 100% dependent on government sources funding money, which is slowly sort of drying. So I think is a very, and I think that is where we have to change our way of thinking, and I think approach companies and philanthropic organizations to provide more money for research in this country. So anyway, this area is a very complex slide I borrowed from a journal article. And in just last 5 years, we have so many small molecule modulators available now. Some of them are in early development, some of them are clinical development, and larger companies like Genentech, Merck, they are all looking at some of this work. And one particular pathway which is described here, I want to focus, and I think I have written the references also and just published in this year. It's a beautiful story that is opening up what we call a STING pathway. STING is a stimulator of interferon genes. All of us know about interferons. We must have seen during COVID time, people were given interferons as a therapy. And these interferon type 1 interferons, they are basically controlled by this particular protein which sits almost at the interface of nucleus and the other compartment, nucleosomes, on the surface of nucleosome. And here, what has been discovered that there is a particular a protein, what we call cyclic GMP AMP synthase, which is in the soluble form, and its function is to recognize double-stranded DNA of pathogen or cancer. So, for example, if you have any, say, COVID infection, or you have a bacterial infection, or you have a cancer, some amount of broken DNA of these invaders will be in your flowing around in your body always. And this G gas, this protein will recognize immediately. When it recognizes, it acquires an enzymatic property, and it start fusing two important small molecule, ATP and GMP, and it start creating this molecule what we call CGAMP. And due to CGAMP, this molecule dimerizes, the G-gas dimerizes and activate this protein called STING. And once you activate this STING protein, immediately it sends the message for the larger production of type 1 interferons. And type 1 interferons are cytokines, and their role is to destroy, like an army, destroy these pathogens. So it's very, very important, and these are internal. Those who are from organic chemistry background can see these are all molecules as a produced by our body as a second messenger. Very, very interesting. And now people have started synthesizing now, designing a small molecule through which you will be able to control. So it's a very new direction that is coming to deal with this particular infections and cancers. So I'm very fascinating with this, and we are starting doing some work in this area in collaboration with a particular pharmaceutical company. And now, very recently, this paper came out, Nature Materials paper, where they're now formulation people design a nanoparticle, how to deliver, because as you see, these are oligonucleotide analog, and some of them are not very stable. So if you can create these nano liposomes or nano delivery method, and there they have shown that by new delivery platform for these, you can actually target in solid tumor. If you don't have those delivery platform, you cannot target go inside the solid tumor. So the next step forward that now we can use these STING agonists using these delivery platform like an RNA molecule. I gave you example. So it's a fascinating new opportunity for designing the vehicles so that we can go to solid tumors rather than the blood cancers. Another area which is very interesting, which I think if there are some plant science people in the audience, I recently gave a lecture that how now we are converging, that plant sciences and animal sciences are now coming together after a lot of almost century or 2 centuries of divergence. And there are fascinating things one is discovering from plant sciences. And one area is very exciting, which is happening right now, that immune system is plant and immune system in our body. Plants have an immune system, by the way, very well evolved immune system, but their immune system is restricted only to innate immune system. Like in our body, we have innate immune system and we have the acquired immune system. Acquired immune system is B cell, T cell based immune system. Innate system is like macrophages, dendritic cells, natural killer cells, and all those. And these are I've just put these two together. If you see left and right, I mean, this is the left is the innate immune system of plant, and right is innate immune system of animal. See, they look same, but at the same time there are subtle differences. And even their ligands, so they're toll-like receptors. So I'm sure you must have heard these toll-like receptors, which are receptors for innate immunity. And I think this is like a gold mine for next generation immunomodulator design, from learning from plant and designing these immunomodulators for human being. So I think that something is nature's secret revealing through this work, and I strongly recommend people to read about this area. Fascinating discoveries are being made, both for therapies and also as an area called adjuvant. And this is one example I'll give you because, as you know, adjuvant is used necessary for any vaccine to work. And most of the vaccines use adjuvant called alum, and we do not know how these adjuvant work till today. We do not know how to design these adjuvant, and that is why this is the area which is totally stopped. But because of this knowledge, now we can systematically start discovering these adjuvant from plant. So this is one example of a adjuvant which was discovered from a plant in Chile, which is a country in South America, where they had isolated this compound called QS21, a saponin. And this is right now, because of COVID vaccine, this is the most sought after molecules, multi-billion-dollar. Everybody is looking for this molecule. And Novavax COVID vaccine is actually using this particular adjuvant for their vaccine, and this enhances both humoral and cell-mediated immunity through innate immunity mechanism, which I talked about earlier. So you can just, if you can grasp that, I'm trying to say that this is a gold mine waiting to be discovered, where systematically correlation of innate immune mechanism of the plant and animal together, and then looking at those molecules which are involved in innate immunity system in plant, can they be repositioned in immune system? After all, we have all evolved together, and maybe billions of years ago, the plant life and animal life diverged, and one came to animal world where we are today, and they went to different direction because of their requirement of the natural environment or natural selection. So it's a fascinating area. I strongly urge people to look into this. I will be very happy to provide you more information and then reading material. Last area. This is for people who are working in microbiology or plant biology. That now this what we call, like all of us know that in our genome, if we look at our genome and we look at our transcriptome, means DNA and RNA, we only transcribe practically 10% of our genome. 90%, we don't know why this is there. 20 years ago, people used to call it junk DNA. You must have heard this term, junk DNA. So 90% is DNA sitting in our body. People, human nature that we don't understand. If you don't understand something, you start saying it is useless. This is a typical human behavior. But now I think we are learning hard way that nature doesn't do anything without any purpose. So now we are understanding that this so-called junk DNA, actually it is silent, and it harbors the memory of billions of years of evolution. And just silently sitting with that information there, and what we call cryptic biosynthetic pathways. They are there in plant, microbes, everywhere. But since microbes are easy to handle, easy to manipulate genetically, work has started happening here. And this area is that where we can now genetically identify that which are the cryptic pathway and which type of molecule it can code, it can produce, but it is not doing. So it has the language, but it is not speaking. Now, can we induce that language to become sort of audible? And that is done by disturbing the genetic makeup, adding, deleting some of the modular PKS pathways. This work was going on in actinomycetes, myxobacteria, endophytic fungus. And now we have reached a stage that we can now uncode that silent cryptic sequence and make next generation new molecules. So in fact, 27% higher rate of discovery of new natural product molecule from the same bacteria. So you have been studying a bacteria for 50 years, but now you sort of create that bacteria to produce next molecule. So it is a very, very powerful technology, which is what some people call it synthetic biology. Some people have a different name. And some of the example I have given here that, now we are converging with the synthetic chemistry and synthetic biology. As you know, when the molecules are very complex, synthetic chemistry, you can make that molecule, but it's essentially useless. That synthesis, for 40-step synthesis, that molecule is never going to be of any use. You just publish it and forget about it. But if you combine these two technologies, you can make those changes. Like here, the balls that I read, these are the changes you can, by tinkering of the genetic machinery of that particular bacteria, and you can make this new molecule. And I've just cited two papers here published, very phenomenal paper. This paper, 2020 paper, where they have whole genetic machinery of colchicine. This is a drug which we use for COVID. And in fact, in CSIR, we are now conducting a clinical trial on this for COVID-19 patients. And then this paper came out, I think, last month or 2 months ago on strychnine, good old molecule. Now they are able to decipher the entire pathway, and they take this pathway and identify, transfer them to yeast, and start producing these molecules. Instead of plant, you start producing in bacteria or yeast. So that is like a future looking like that. When you have very important molecule, for example, you discover from some species, you can start producing other species, and that's where this 90% of genome of microbes, which that imagine that 10% of the genome discovery or gave us so many fantastic antibiotics. Now, what in the future, this when this 90% is able to be decrypted, how many more antibiotics, how many more therapeutics we will be able to discover? So it's a fascinating new field that is opening up. Initially, it took some time for basic biology, almost 20 years. Now it is in the translation phase where this is now giving us practical and real therapeutics. And in this context, there is one interesting aspect that you can actually kickstart these cryptic pathways by using epigenetic modulator. So as you know, epigenetic modulator, if you disturb the epigenetics, that also changes the composition of the cell. So this is our own work that we did, and there's several work in this direction is going on. For example, this is well-known molecule on the right, which is produced by this fungus. They are known for 50, 60 years. But once you treat this culture with a epigenetic modulator like valproic acid, you see on the HPLC how the composition of molecule drastically changes. Some molecules have disappeared, some have appeared, some have become some smaller, and you can see how their yield into the composition has changed. So you can imagine, you can take the same strain on which you are working, but you can now create new molecule from this. And valproic acid is one example. There are hundreds of epigenetic modulators. So if you cannot do genetically, you can do epigenetically, which is much easier. Flask, small molecule, culture, HPLC, purify, characterize. So I think that's also is just one, and there's a whole lot of literature that is coming up. This was work that we did almost 4-5 years ago, but after that, a lot of things have happened. So I think these are some of the possibilities that are emerging, and these are some of the examples I gave about what is my view of these areas. And of course, people, my colleague, whatever little that we did, I just gave example that what is happening globally. Our contribution is very minimal, but at least some contribution we made in this area. And say these are my colleague at my Jammu Institute, whom I'm eternally grateful for doing phenomenal work, and large number of PhD students. And we could do this substantial work, which was published in all over the literature from top chemistry, top biology journals. So I think this is the advantage of working on these areas, and your work is more visible and productive. So I thank you all for listening to me carefully. Hope I have stayed with the time, and if there is a provision for questions, I'll be very happy to take some of those questions. Thank you.
Thank you, Professor Viswakarma, for a wonderful lecture. I will ask the audience for just 1 or 2 questions.
My presentation is gone?
Yeah, presentation is very nice. Just
Okay. Yeah.
Yeah, it's an excellent talk. I have just one query from you. How do you envisage the role of theoretical and computational biologists in addressing these problems in the future?
Yeah. So it's a very, very good question. I tell you, these are, and India particularly is, as you know, is reasonably strong in these areas. And that one example I gave you about this deciphering these cryptic gene clusters, cryptic gene clusters in biology. And since we had the scientific community had just started, so they started the bacteria. But imagine if we look at the human genome, where we have 90% of that DNA which is sitting there, which for a long time we called sort of junk DNA. Now with this technology, and there are companies which have now developed software tools to actually predict. So computational biology is actually the future. And secondly, with the arrival of the AlphaFold and all these resources, I'm sure AlphaFold is a revolution that we are witnessing. I think it is, to me, as important as the DNA discovery in 1953. It is going to change. Every student can now get the structure of every damn protein that is there in human biology. And I think that's a challenge for computational biology. As a humble chemist, I can't address that complexity. So I think these are the challenges for also in protein-protein interaction area. This is essentially a computational biology problem. How these protein-protein surfaces interact, how they change in the biological cells. It's huge, huge opportunity. And I keep discussing these areas with Dr. Sekhar Mande, who is a computational biologist, the former DG of CSIR, and we keep discussing with him. So I think it's an excellent question. But I'm not a computational biologist, so I cannot give you very specific.
Because whatever you are showing that small molecules, as a theoretical chemist, I know this is quite large for me to calculate accurate data. So that's why I was asking how to handle such large systems with reasonable accuracy, to generate acceptable data for the experimentalists. So that becomes a very challenging problem for even probably for computational biologists as per the resource available as on today.
Yeah. Thank you.
There is one question from an online participant, Mr. Ajay Kumar Sahu. His question is, why kinase inhibitors are generally covalent inhibitors?
Why kinase inhibitors?
Are generally covalent inhibitors.
No, that statement is not actually correct. Most of them are not covalent inhibitor. Most of them, but there are some. And now there is a greater trend towards designing covalent inhibitors. For a long time, it was considered that they are not good because they will sort of covalently link to protein and create immunological reactions and other things. But now I think, in fact, very, very interesting. Today's issue of Nature Review Drug Discovery has a comprehensive review on covalent inhibitor design for therapeutic discovery. But the statement that most of the kinase inhibitors are covalent inhibitors is not correct. But there are some which are, yeah. But the answer is that why that answer is not there. I don't know, and perhaps at least I don't know. Yeah.
So, Professor Viswakarma, I enjoyed your talk. I have also a small query. See, when you showed these kinase inhibitors, actually, if in case of you see the bacteria, you are actually phosphorylating is carboxylic acid like aspartic acid and glutamic acid, carboxylic acid. So essentially, it is a mixed acid
Anhydride, yeah.
and I wonder how these anhydrides are stable in a biologically environment where a lot of water is there. Are they getting folded into this hydrophobic pocket so that they are getting stabilized or any other?
Absolutely. Histidine and aspartic acid. These are anhydride basically.
Yeah.
So very important question. But this bacteria and primitive life, most of the primitive life, bacteria, archaea, they use this mechanism. And then in our system, we don't use those. So I think this is a very interesting question. So they are essentially pocketed inside these pockets so that they are protected for whatever the purpose. So why I showed this, that now there is an effort to design next generation antimicrobial compounds targeting these proteins. So now imagine the proteins which are phosphorylating these things, how weird those proteins would be, right, in bacterial system, and how different they will be from human proteins. So it's a very important point you noticed, and I'm happy that my purpose of putting there bacteria versus human was useful.
So this last part of your lecture is very interesting. It is reminding me a very pathetic future about synthetic organic chemistry. You know, in about 2 years back, there was a publication came in Untitled International Edition, where they say that now the chemistry will be the synthetic biology.
Yeah,
Okay.
I agree with that. I tell you. So it is high time.
Will be the organometallic chemistry.
Sorry?
That organometallic chemistry involving transition metals.
Yeah. Organic, see, if you see organometallic chemistry, nature does fundamental organic metallical chemistry of its own. See,
Yeah.
many of metalloenzyme, now I think time has come to combine both these inorganic metallochemistry and organic metallochemistry together and then understand these systems. So I think it's a very fascinating, and I think the message for us in India is rest of the world has got that message that we have to make extra effort to work together. Chemists should try to at least talk to a biological person or something. I think we can do bigger things rather than we are doing in isolation. That's a message from these directions. Yeah.
So, Professor Viswakarma, in your lecture, you have given a very interesting information to both the biologists as well as the people who are working in the chemistry field. So, I think all the participants have enjoyed your lecture, both by those who are present here in person as well as online. And I think your message will be highly benefited in their future research career. Okay, and with this, I thank you very much for accepting our invitation and giving such an exciting lecture.
Thank you. Thank you.
On behalf of HB and I, we cannot deliver you the book posted to your place, but we would like to show it to you.
Yeah.
Yeah. Okay, so it will be delivered to your at your address. Okay, and again, thank you very much for spending your valuable time and giving good.
And my apology, but when I come to Bombay next time, I will visit you. I will visit you. Yeah.
Yeah. Thank you very much.
Thank you. Thank you.
Thank you. Bye.